back, Biology 115, to the genetic basis of cancer, a brief summary, and I really mean it when I say brief. So after this lecture, you should be able to compare and contrast the terms proto-oncogenes, oncogenes, and tumor suppressor genes. What do each do? What does each mean? Explain why multiple mutations are necessary before cancer occurs. Okay, so the first thing we're going to tackle is these terms. So proto-oncogenes, proto means before. So these occur before oncogenes. When these become mutated by UV light or some carcinogen, they become oncogenes. So proto-oncogenes we really need because they do normal sort of daily things. They make these proteins that we need to survive. So that's what they're doing normally. They're making these proteins. When they become mutated, they become oncogenes, and oncogenes are what actually cause cells to become cancerous. So remember, cancer is undivided cell growth. So when a cell becomes cancerous, it divides uncontrollably. Nothing is stopping it. And can divide forever and ever and ever with no stops. Okay, and just as a quick refresher, the 3 stop signs in the cell cycle to check the DNA is G1S, G2M, and then the M phase or spindle checkpoints. Those are our 3 checkpoints. 1, 2, 3. They are named for the phases between which they occur. So G1S occurs in interphase between G1 phase and S phase. G2M occurs at the end of interphase, the beginning of prophase. And M here stands for the mitotic phase, where the nucleus actually divides. And then the M spindle checkpoints make sure that all of the chromosomes has their centromeres attached to the spindle and lined up at the center of the cell. And each one of these checks the DNA in a different way. Okay, so those are our stop signs. These are our checkpoints. So if these are not functioning, the cell just goes and goes and goes. So a favorite analogy for these proto-oncogenes and oncogenes is like a gas pedal on the car. So you normally need these proto-oncogenes, but when they become mutated and something happens like perhaps too many copies, or they switch the places with something else and it gets activated too much, it's like pressing down on the car's gas pedal and it just goes. So this causes oncogenes, which are bad. And then because these oncogenes are what causes the cells to divide rapidly. Tumor suppressor genes you can think of like the brakes, because their job is to suppress tumors. So what suppress means is stop tumor formation. So these are like the stop signs. So these are like the brakes on a car, and this is like the gas. So if some proto-oncogene gets mutated to an oncogene, it can still be stopped by tumor suppressor genes, because those tumor suppressor proteins can lay on the brakes and stop these tumors from growing. So they're suppressing the tumors. So the way tumor suppressor genes works is through apoptosis. So what apoptosis is that programmed cell death. So that's the real difference here. So these genes make proteins that see that this cell is out of control. It's going to become cancer. So it stops that by destroying the cell. And so this is really where the cell checkpoints come in, the G1S, the G2M, and the M spindle phase. Because if we don't stop at these checkpoints, the tumor suppressor genes can't do their jobs. So if the tumor suppressor genes get mutated, they become inactive. So, as you can imagine, if the thing that causes cell death becomes inactivated, then it's going to cause cancer. So you actually need a combination of mutations from tumor suppressor genes and proto-oncogenes before you can actually get cancer. So mutants are inactive, and then that means the cells can't die. Cells can't die, and that's what cancer is. So they're just going to divide rapidly. Okay, and that's it.